on their structure and function. One interesting area play a role in various processes and diseases. Stem cells, heart cells, and more. Intermediate filament-associated diseases are a group of disorders characterized by mutations in genes encoding intermediate filament proteins, leading to the disruption of the cytoskeletal network. One prominent example is epidermolysis bullosa simplex, EBS, a genetic skin disorder caused by mutations in keratin genes. The loss of structural integrity in the epidermis leads to blistering and extreme vulnerability to minor trauma. Similarly, mutations in neurofilament or vimentin genes have been linked to neurodegenerative disorders such as amyotrophic lateral sclerosis, ALS, and Parkinson's disease. In the context of neuronal development and regeneration, intermediate filaments play crucial roles in providing mechanical support and guiding neuronal migration. Neurofilaments, which are intermediate filaments specific to neurons, contribute to the structural stability of axons. Additionally, intermediate filaments are involved in the axonal transport of important molecules required for neuronal growth, maintenance, and synaptic function. Their dynamic regulation during neuronal development and the regenerative process highlights their significance in neural plasticity. Cellular aging, also known as senescence, is characterized by the accumulation of various hallmarks, including altered morphology, impaired cellular functions, and increased susceptibility to stress. Intermediate filaments, such as vimentin, desmin, and lamins, play crucial roles in maintaining cellular architecture and protecting cells against mechanical stress. However, during aging, the expression and organization of intermediate filaments undergo changes, leading to altered cell mechanics and impaired cellular functions. Understanding the relationship between intermediate filaments and aging is essential for unraveling the mechanisms underlying age-related diseases. Stem cells possess unique properties, including self-renewal and the ability to differentiate into various cell types. Intermediate filaments contribute to the maintenance of stem cell pluripotency and regulate cellular organization within stem cell niches. For instance, cytokeratins are important components of the cytoskeleton in epithelial stem cells, helping to maintain their regenerative capacity. Additionally, intermediate filament proteins have been implicated in the regulation of stem cell fate decisions and differentiation processes. During tissue repair and wound healing, intermediate filaments dynamically reorganize and function in cellular migration, adhesion, and wound contraction. In epithelial cells, cytokeratins enable cells to regain connectivity and restore tissue integrity. Moreover, intermediate filaments contribute to the alignment and strengthening of fibroblasts within the wound bed, facilitating the deposition of extracellular matrix components necessary for tissue remodeling. Understanding the intricate mechanisms underlying intermediate filament dynamics in wound healing can offer insights for developing therapeutic strategies for promoting tissue regeneration. Intermediate filaments have been implicated in the regulation of immune cell function, including mechanisms involved in migration, adhesion, and antigen presentation. For instance, vimentin, an intermediate filament protein, facilitates the migration of immune cells towards sites of inflammation through interactions with molecular motor proteins. Intermediate filaments also participate in the assembly of antigen presentation complexes in dendritic cells, playing a critical role in immune surveillance and activation of adaptive immune responses. The integrity and mechanical resilience of cardiomyocytes, the muscle cells of the heart, heavily rely on intermediate filaments. Desmin, a specific intermediate filament protein expressed in muscle tissues, forms a network that stabilizes the sarcomeres and links them to the cell membrane. Disruption of desmin filaments impairs the contractile function of the heart and is associated with cardiomyopathies and heart failure. Understanding the role of intermediate filaments in heart muscle cells is vital for designing interventions to prevent and treat cardiovascular diseases. Apart from their structural functions, intermediate filaments play a role in the regulation of gene expression. Intermediate filament proteins, such as nuclear lamins, contribute to nuclear organization and chromatin architecture, influencing transcriptional regulation. Additionally, intermediate filament-associated proteins interact with transcription factors and modulate their activity, providing a link between the cytoskeleton and the regulation of gene expression. Intermediate filaments can influence cell signaling pathways by serving as platforms for the recruitment and organization of signaling components. For example, scaffolding proteins called plectins link intermediate filaments to signaling molecules, facilitating their activation and downstream signaling events. These interactions contribute to various cellular processes, including proliferation, survival, and differentiation. Elucidating the intricate details of intermediate filament-mediated cell signaling is pivotal for comprehending cellular responses in health and disease. The intermediate filament network interacts with the extracellular matrix, ECM, and contributes to tissue organization and mechanical integrity. Intermediate filaments can physically attach to ECM components, such as fibronectin and collagen, facilitating cell ECM adhesion and migration. Through these interactions, intermediate filaments also participate in the mechanotransduction process, converting mechanical cues from the ECM into biochemical signals that regulate cell behavior and tissue homeostasis. Neurodegenerative diseases, including Alzheimer's, Huntington's, and Parkinson's disease, are characterized by the progressive loss of specific neuronal populations. Intermediate filaments, particularly neurofilaments, are involved in the formation and maintenance of axonal structures. Alterations in intermediate filament assembly, turnover, or degradation have been implicated in the pathogenesis of neurodegenerative diseases, contributing to axonal dysfunction and neuronal degeneration. Mechanotransduction refers to the conversion of mechanical forces into biochemical signals, influencing various cellular processes, including proliferation, differentiation, and migration. Intermediate filaments act as mechanical stress sensors, transmitting and distributing forces throughout the cell. These forces, in turn, can influence key signaling pathways and modulate cellular behavior. Understanding the role of intermediate filaments in mechanotransduction provides insights into cellular responses to physical cues, which is relevant in cell biology and tissue engineering. To support, organization, and regulation, repair, immune cells, physiology, cellular matrix, neurodegeneration, and mechanobiology is vital for medical professionals to improve